And so the level of virus. In this last immunology lecture, I'm talking about how the immune system controls the outgrowth of tumor cells. And that is because in our bodies, we always have mutated cells growing out. If later on in your careers, if any of you ever get into a laboratory and work on tissue culture, you'll always find that when you're growing cells outside of the body, you will always notice that some cells, eventually you get mutations happening with some cells, and that they behave differently from the way they were grown initially. So the objective of this lecture is to define the properties of cancer cells and the immune responses directed against them, to highlight the interaction between this immune monitoring of the outgrowth of these cancer cells and how these cancer cells that can escape the immune response can then break out to develop a full-blown tumor. So to define these immune evasion mechanisms used by tumor cells, to define the targets that are recognized by the immune system expressed on these tumor cells, and then what's a big topic at the moment is new developments in manipulating and amplifying the immune response to control tumors. So first of all, you've already, Dr. Barnes covered this with you on tumorigenesis, that in tumor cells there's a breakdown in this control of cell cycle regulation, so that cell growth is amplified and cell death is reduced. So you get this imbalance in cell proliferation. And you know that this process involves a number of mutations, whether they're in cell cycle genes or in the BCL apoptotic pathway and so on. And the initial observations of immunologists in the early 50s and even earlier was that the immune system had a role in controlling the outgrowth of these cells. And these early observations, when this was in the beginning of the 20th century, was that some patients who had sarcoma, if they had an additional infection with a bacteria like Strep pyogenes, this sarcoma went away. And so these clinicians then reasoned that maybe by stimulating the immune response against the bacteria, those stimulated immune system, could then attack the sarcoma tumor cells. And so they did these rough clinical trials with people who had sarcoma. They deliberately gave them Strep pyogenes and Serratia marcescens bacterial infection to try to see if by stimulating the immune response against the bacteria that might have some effect on the tumor. And some patients seemed to show some regression in the sarcoma, but it wasn't statistically significant enough to carry on with those sort of trials. Nevertheless, then pure immunologists in the laboratory found that they could induce tumors in mice either by painting chemical carcinogens on the skin and inducing the tumor, and then taking the blood and looking at the reactivity of these lymphocytes. And from that, they could identify particular, what they call tumor-specific antigens as a result of these artificially induced tumors. And these early experiments in the 60s, they could show that, for instance, what they did was induce a tumor in a mouse, extracted that solid tumor, and again, standard procedure, you radiated these cells, not giving them enough radiation to kill the cells, but to keep them alive, so they were there as targets. And then if you injected them into a mouse, and you found that, you injected these irradiated cells into a mouse, and then after a few weeks, within the same mouse, you irradiated, non-irradiated, the same tumor cells, but not irradiated this time. So these cells had the potential to grow up into tumors. But when they injected these viable live tumor cells into these mice, no tumors have developed. So the conclusion there was that that initial injection with the irradiated cells was similar to a vaccination. You induced an immune response against these tumor cells, so that on secondary injection, when you gave live cells, the activated immune response could kill those cells directly. And this was specific because if you gave a second injection with a different tumor, the different tumor cells developed into a tumor. So there was antigenic specificity in this experiment. So from these studies, they found that the primary source of these immune reactive cells were cytotoxic T cells. So the conclusion is that cytotoxic T cells, in recognizing some particular tumor-specific antigen presented on class 1 MHC on these tumor cells, are responsible for controlling the outgrowth of tumors. So obviously, if you have a tumor cell that's developing, it has to be taken up by a dendritic cell through cross-presentation, degraded so that the tumor-specific peptide can be presented on class 1 for CTLs and on class 2 for T helper cells, and then you get a effective CTL response. But the fact that cancer is relatively common means that this process of tumor surveillance isn't 100% effective. And so then immunologists try to reason why is this. So the further studies showed that these tumor antigens weren't that immunogenic, that in many cases these tumor cells can escape the immune response because they're weakly antigenic, and this is reasonable considering that the tumor is derived from the body itself, so many of these antigens could be similar to self-antigens. But other studies also showed that tumor cells, through the mutation process, develop many mechanisms for evading the tumor, for evading the immune response through direct mechanisms. So based on this, they revised this concept of tumor surveillance to say that there's a dynamic process that occurs between the initial development of a tumorigenic cell and a process whether this tumorigenic cell can survive and then break out into full-blown tumor or be controlled. And this idea is that this relationship between the immune response and the tumor cell involves 3 phases. And the first phase is what we'd have in a normal, in all of us, where there's an elimination phase, where the tumor surveillance mechanisms are sufficient to kill any beginnings of a tumorigenic cell proliferating to significant numbers. Over time, you have this equilibrium phase where most of those tumor cells will die, but some will survive that immune response. And so this is similar to any type of host-pathogen relationship, whether it's a virus-infected cell, bacteria, or whatever. The ones that survive the immune attack have evolved to be able to evade the immune response. And so if you get this shift in the balance, where you get enough of these tumor cells breaking out, because they're highly proliferative, then they can increase in numbers so much that the immune response can no longer counteract them. And so they can break out into full-blown tumors. And this is what's called the escape phase. And the evidence for this equilibrium phase, this continuous balance between immune surveillance and survival of the tumor, comes from two observations. One is that about 5 years ago, there was a patient who, in a transplant operation, there was a donor whose kidneys were used for kidney transplant for 2 patients. And this donor had had a melanoma about 12 years back, which was successfully treated. But 2 years after receiving these donor kidneys, both patients came down with the same melanoma that the donor had experienced. So the idea was that even though the donor had survived the melanoma, there were melanoma cells hanging around in her body, but at low levels that were controlled by the immune system. And some of these might have resided in those kidneys, so that when they were transplanted into the recipients who were receiving immunosuppressive therapy, these melanoma cells broke out to develop cancer. The other is that there's in people who get other transplants and are undergoing immunosuppressive therapy, a lot of them get B-cell lymphomas as a result of EBV infection in these patients. So I told you before that EBV has a propensity to infect B cells and cause transformation in these cells. But in normal circumstances, our cellular immune response controls that virus. When we get immunosuppression, then the virus can break out and transform the B cells to cause a leukemia. Don't bother learning this slide, but this is just an illustration from studies of knockout mice, so where they delete the genes in these mice for particular components of the immune system. So in these knockout mice, where they've knocked out perforin, what activity are they knocking out in these mice?
Cytotoxic T cells and NK cells.
Right, cytotoxic T cells and NK cells. And they find that these mice, a lot of these mice come out with lymphomas. So this gives an idea of the role of this component of the immune system in controlling these types of outbreaks. With RAG1 and STAT1, with RAG and STAT1,
all
the RAG gene,
antibodies and the
BDJ and STAT1 is signaling. So essentially you're creating skid mice or severe combined immunodeficiency mice because they've got no T cells, no antibody producing B cells. And in these mice, you find lots of gut epithelial and breast tumors. With gamma delta T cells, this other component of T lymphocytes with this gamma delta receptor, you have no interferon production and you find skin tumors. So this just gives some indication of the roles that these components of the immune system play. But in mice, so it might be a different situation in humans, but nevertheless it gives you some idea of how these immune components do in controlling these particular tumors. Regarding the targets, the antigenic epitopes that are recognized by cytotoxic T cells, they're grouped into two main types. So we have tumor-specific transplantation antigens and tumor-associated transplantation antigens. Don't get confused with this word transplantation antigen. It's not to do with organ transplants, but it was used by the immunologists who were basically transplanting tumors from one mouse into another to look at those antigens, right? But this is in the context of tumor development. So specific tumor antigens. So these are antigens which are, through mutations in the tumor cell, are specific for the tumor, not for normal cells, right? Tumor-associated antigens are antigens which we also find in the normal cell, but because the tumor cell has dysregulated the control of these antigens, they're overexpressed in the tumor situation. So they're essentially normal cellular proteins, but changed in terms of their expression control. Right, so that's an important difference to remember. So regarding these tumor-specific antigens, through gene sequencing of a lot of tumors, they find that the longer the tumor goes, the more mutations there are in the genome. Many of these mutations in the DNA sequence are irrelevant to the tumor. They're just what they call passenger mutations. So they have no real function or bearing on the antigenic specificity that might be recognized by the CTLs, or any effects on controlling the growth of the tumor. But nevertheless, they're called neoantigens because they are altered self-proteins compared to the normal situation. But also remember that even though they're neoantigens, that they, based on the amino acid sequence, they need to have the correct amino acid sequence to be able to be loaded onto self-MHC class 1s. So a lot of them, through the amino acid sequence, won't be, because they don't have the right sequence, or the right fit. But nevertheless, you can find some particular gene lesions, mainly associated with cell cycle control or signaling, such as RAS, MYC, cyclins, and again, I'm sure Dr. Oban's covered that with you, right? These are called oncogenes. Other types of tumor-specific antigens are carried by particular oncogenic viruses, like the papillomavirus for the E6, E7 protein, or the HTLV1, human T cell lymphomavirus. And then tumor-associated antigens, like I said, these are normal proteins, but the control of these proteins have been dysregulated. And the prime example of these are these melanoma-associated antigens called MAGE, which are actually proteins that you find in the testes. Because during embryology, during the development of the testis in the male embryo, these proteins are expressed in the development of the testis, but with the as that embryo matures, then transcription factors control to stop production of this protein. But because of this dysregulation in the tumor cell, particularly in melanomas, then they start expressing these embryonic testis proteins. Similarly, with liver cancer, one of the common tumor-associated proteins to be expressed is alpha-fetoprotein. So that's a common biomarker for liver cancer cells. Another well-studied protein is in the breast cancer, which is a homologue of the EGF receptor, and this is HER2/neu, which is a variant of ERB2, which is a tyrosine kinase signaling protein for the EGF receptor. And so in these breast cancer cells, there's overexpression of this receptor, which means that they are stimulated more strongly to EGF, resulting in the proliferation of these breast cancer cells. Other modifications of normal cell components is mucin, this glycoprotein, which has a different type of glycosylation pattern compared to normal cells, and that also improves the oncogenicity of these cells. Now, in terms of the tumor evasion mechanisms of tumors, there's several strategies that tumor cell has for evading the immune system. First of all, like I said, because even though it's tumorigenic, these self-peptides still have high homology to normal self-peptides, so they have low immunogenicity. Because of that, if a dendritic cell takes up these tumor cells and processes them by cross-presentation, the immunogenicity of that antigen isn't enough to stimulate a full-blown activation of the dendritic cell, which means that it's not going to be expressing co-stimulatory molecules, cytokines, and whatever, which means that it's not going to stimulate a naive T cell reactive to this antigenic epitope to become a full-blown effector cell, right? The other thing is that many tumors express these negative regulatory signaling molecules like PD-1 ligand, CTLA-4, so they can block CD28 activation of these CTLs. A third aspect is that, as I said, because as the tumor progresses, it undergoes many mutations in the genome sequence, and so this can also change its antigenic epitope expression, its antigenic epitopes. And then a fourth is that many of these tumor cells can either express these immunoregulatory cytokines IL-10 and TGF-beta, or they can promote the outgrowth of these tumor-specific antigen Treg cells, so blocking cytotoxic specific tumor activity. And then the fifth type is that when you get a solid tumor, you usually get these collagen capsule around the tumor, and that provides a physical barrier for penetration of CTLs to get into that tumor mass or for antibodies to get in. Related to this idea of the solid barrier is when a tumor, when some tumor cells start to metastasize, so they get out of that primary tumor into the circulation, they get coated with platelets, and this protects them from attack by CTLs, which allows them to then circulate through the body, break out of the circulation into other types of tissues. So now we come to the advances in modulating the immune response for immunotherapy. The first is adoptive T-cell therapy, which involves taking blood from these tumor patients and trying to isolate their tumor-specific CTLs and culturing them with IL-2 in vitro. So you expand them in numbers and then inject them back into the patient. Another type is in a similar type of strategy. I was involved once in a clinical trial where we had a patient who had advanced liver cancer because of hepatitis C infection. And what they did was they did a resection of his tumor liver, and we took the liver offcuts and mashed them up in the laboratory, and we took his blood and expanded his dendritic cells. We then fed the dendritic cells with the liver cancer homogeneous. So the idea then was that the dendritic cells would take up the tumor antigens from the liver cancer and then present any hepatitis C related tumor antigens on class 1 MHC, and then injected these DCs back into the person so that these would be presenting the tumor antigens to the CTLs for attacking the other parts of the liver tumor. And there was some regression in the tumor, so it did work. So that's another type of therapy. The one that has brought the biggest interest in the past 5 years is this genetically engineered receptor called a chimeric antigen receptor, or CAR. And what this involves is getting the gene sequence for the epitope recognition of the FAB region of the antibody. So this is the CDR 1, 2, and 3 gene sequence. cells and fusing that onto the signaling chain of a T cell receptor. So in this way, you've got a hybrid of an antibody recognition site linked to T cell signaling portion of a T cell receptor. And then having that sequence, so in essence, you're creating an artificial gene which codes for antigen recognition based on an antibody molecule that can signal via T cell CD3 signaling. You insert that into a vector like a retrovirus and then use that to infect a CTL. The messenger RNA for this chimeric receptor gets expressed on the CTL. So now you have a CTL which can recognize antigen on your target cell without needing class 1 MHC presentation. So you can use it for everybody, right? And this has been really successful in many tumors.
What about, doesn't it need still the co-stimulatory effects?
No, because the recognition is just through FAB binding. And that recognition, similar to the way an antibody surface receptor works, is sufficient to transduce the signal through CD3. So it doesn't need class 1, it doesn't need co-stimulation or anything like that. The other big breakthrough is the so-called checkpoint therapy. I talked about checkpoints yesterday in regards to tolerance, right? So the two checkpoints you have are, remember, CTLA4, which is the suppressive part of CD28, both recognized in B7, and PD1 ligand. So what they do is they give these patients, because tumor cells can try to evade the immune system by promoting CTLA4 expression on the reactive CTLs. So what they do is to give antibodies against PD1 or to CTLA4. And so this blocks the suppressive activation. So only CD28 on CTLs can recognize B7. So you get hyperactivation. And this has been shown a lot of regression in tumors. The third strategy is to give monoclonal antibodies against particular growth factors such as HER2, the EGF receptor for breast cancer. So then you're preventing EGF from stimulating these growth factor receptors on the breast cancer cell. And then obviously the fourth strategy for the tumor-specific antigens carried by viruses is through vaccination against hepatitis B virus or papillomavirus. Yeah, that's what I've just given here. Okay, is that it? Any questions?